Organogenesis in plants. So it is the basic concept of organogenesis in plants. If we take plants, from root to aerial shoot system, we take parts of plant as a explant. If we used explant for the culture of in vitro or in aseptic conditions, that culture is called indirect organogenesis. If we used plant parts, large number of tissues, mean branches of shoots, like seeds, they are developed into complete plantlet by the direct organogenesis. Direct organogenesis, we use large amount of tissues from the parental plant or seeds from the plant. They develop into complete plantlet. This process is called direct organogenesis. During the direct organogenesis, we take shoot part, the shoot plantation in the soil. The shoot terminal part contain meristematic tissues. It produce auxins. The auxins can generate root system. It develop into complete plant by the direct organogenesis. If we take explant from these parts of plant, the expand transfer into the nutrient medium, it develop into callus. The callus is added with the growth hormones in different ratios. They develop into sometimes root organogenesis, sometimes shoot organogenesis. Sometimes somatic embryos develop into complete plantlet. So this total 3 ways are belongs to indirect organogenesis. Now let's discuss about the detailed explanation of plant regeneration pathways. Plant regeneration pathways. Here it is. First we discuss about introduction of organogenesis. Plant tissue culture. Plant tissue culture is a collection of techniques used to maintain or grow plant cells or tissues or organs under sterile conditions on a nutrient medium of known composition. Plant tissue culture is widely used to produce clones of a plant in a method known as micropropagation. The common pathways of plant tissue culture regeneration are: first one is propagation from pre-existing meristem, like shoot culture or nodal culture. Second one, organogenesis. Third one, non-zygotic, means somatic embryogenesis. And fourth one, zygotic embryogenesis. So these are the common pathways of the regeneration of plants. Now, organogenesis is our topic. A plant contains many organs like meristem, cortex, phloem, epidermis, or consist of structural unit called cell. Because an cell have to nature of create whole plant, like any organ or tissue of plants also show some nature, mean they also create to whole plant in vitro condition. So the basic cell, the structural unit of organs called cell. The cell create or produce whole plant or any organ of or any tissues of plant in naturally. If plant organ used in vitro condition to generate new plant, this process is called organogenesis. A definition of organogenesis. The development of adventitious organs or primordia from undifferentiated cells mass in tissue culture by the process of differentiation is called organogenesis. The development of adventitious organs or primordia from undifferentiated cell mass in tissue culture by the process of differentiation is called organogenesis. So undifferentiated mass of cells develop into differentiated mean root and shoot system is called organogenesis, or the formation of root, shoot, or flower buds from the cells in culture in manner similar to adventitious roots or shoot formation cuttings is called organogenesis. The formation of roots, shoot, flower buds from the cells in the culture in manner similar to adventitious roots or shoot formation in cuttings is called organogenesis. First, organogenesis is coalogenesis. Meaning of coalogenesis, the type of organogenesis by which only adventitious shoot bird initiation take place in the callus tissue. Rhizogenesis, a type of organogenesis by which only adventitious root formation takes place in the callus tissue. If we produced shoot is called coalogenesis. If we produced root is called rhizogenesis. It is also one type of organogenesis. Now, types of organogenesis. In plant tissue culture, undifferentiated tissues are referred to as callus. Although a callus can contain meristematic nodules that may not be obvious to the naked eye, but which never developed further unless sustainable or suitable conditions are supplied. Suitable conditions are supplied. Development of organized structures can follows one of the three pathways. One is shoot regeneration based on a unipolar structure with a shoot apical meristem. Second one, root regeneration, essentially a unipolar structure with a root apical meristem. And third one, somatic embryogenesis, in which there is a bipolar structure. First, we discuss about indirect organogenesis. Plant organ formation on callus tissues derived from explants. Plant growth regulators and differentiation. The classic observation of Skoog and Miller that the direction of differentiation could be influenced by the ratio of exogenously supplied growth regulators like auxins and cytokinins. They observed in tobacco stem pit culture the high ratio of auxins and cytokinins that lead to initiation of roots, whereas a low ratio lead to development of shoot. And although there are many species for which this sample manipulation will not work, in general auxins, like example, indole acetic acid, naphthalene acetic acid will stimulate regeneration of roots. And cytokinins example, BAP, benzyl aminopurine, will promote regeneration of shoots or embryos. Now next, direct organogenesis, formation of organs directly on the surface of cultured intact explants. The process does not involve callus formation. The role of growth regulators, direct organogenesis bypasses the need for a callus phase. A good example is the formation of somatic embryos. Most evidence suggests that direct embryogenesis proceeds from cells which were already embryogenetically and competent while they were part of original differentiated tissues. Factors affecting the organogenesis. In vitro organogenesis is controlled by a number of factors other than phytohormones. Phytohormones, nothing but phytohormones. Such factors are discussed below. First, we discussed about the size of explant. Organogenesis is generally dependent upon size of explant. The large explant consists of parenchyma, vascular tissues, cambium, have greater regenerative ability than the smaller explant. Second factor is source of explant. The most suitable part of the plant for the starting culture will depend on species. Leaves and leaf fragment many plant species like Begonia, Solanum, Nectonia, Crepis, and others have shown capacity to regenerate shoot buds. Age of explant. Physiological age of explant is important for in vitro organogenesis. In Nicotina species, regeneration of adventitious shoots is only noted if the leaf explant is collected from the vegetative stage, that is before flowering. Seasonal variations, bulb scales of Lilium speciosum. Regenerate bulblets freely in vitro when explant is taken during the spring and autumn period of growth, but same explant collected from the summer or winter season does not produce any bulblets. Fifth point about factors of organogenesis, oxygen gradient. In some cultures, shoot bud formation takes place when the gradient of available oxygen inside the culture vessels is reduced, but rooting requires is high oxygen gradient. Quality and density of light. The blue region of spectrum promotes shoot formation and red light inducing root. Temperature. Most tissue culture are grown successfully at temperature around 25 degrees Celsius. In number of bulbous species, optimum temperature may be much lower than about 15 to 18 degrees Celsius. And culture medium. Culture medium solidified with agar favors bird formation, although there are some reports about the development of leaf shoot birds on culture grown in a liquid medium. The pH of medium. The pH of culture medium is generally adjusted between 5.62 or 5.6 to 5.8 before sterilization. The pH may have a determining role in organization. Ploidy level. Variation of chromosome number, that is aneuploidy, polyploidy, and others of plant cells in culture has been well documented with the increases in chromosome instability. There is a general decline in a morphogenetic potentiality of the callus tissue. And age of culture, engage of engage of culture frequently produce organs, but the organogenetic potential may decrease and ultimately disappear in old culture. Application of organogenesis. Plant tissue culture or organogenesis now has direct commercial application as well as value in basic research into cell biology, genetics, and biochemistry. Micropropagation using meristem and shoot culture to produce large number of identical individuals. Screening program of cells rather than plants for the advantageous characters, large scale growth of plant cell in liquid culture as a source of secondary products, removal of viruses by the propagation for meristematic tissue. Finally, conclusion about the organogenesis. Organogenesis is the development pathway in which shoot or root have been induced to differentiation from a cell or callus cluster. Induced to differentiation from a cell or callus or cell cluster. In vitro plant regeneration by organogenesis usually involves induction and development of shoot of explants tissue. Shoot from the explant tissues, development of shoot from the explant tissues. It is the organogenesis definition and their types and their factors and conclusion of organogenesis.